information from the two eyes converge or go to equivalent places within the brain. We saw that for much of the visual field, information could be detected by both the right eye and the left eye. And this brings up the interesting question of how, in these binocular regions of the visual field, information is transmitted to the two sides of the brain. We saw that the first way stop on the main visual pathway is, of course, at the lateral geniculate nucleus. We saw that the primate LGN is made up of 6 layers of cells. It's not the way in all animals. The cat has 3 layers, for example. The rat has no layers at all. Other animals have different systems. It's actually very interesting from an evolutionary point of view, because it appears that there's so many different layering systems that this is something that appears to have evolved separately in different groups of animals, which is a really interesting thing in terms of brain evolution. There's clearly something useful about having the LGN laminated, but different animals have come up with different ways of doing it. We saw that of the 6 layers, 3 get input from one eye and 3 get input from the contralateral eye. So although both the right and left eyes project to the lateral geniculate, there's total segregation of right and left eye input within the LGN. We saw the 2 eyes then, the 2 retini project to the LGN, and the LGN on each side projects to the visual cortex, area 17 on the same side. It projects to layer 4 of visual cortex, and we saw that this segregation continues. So looking at the cortex, this is an animal from which a tracer has been injected into one eye under conditions, I hope you recall, that allow for transsynaptic transport. And you can see that looking at this cross-section through visual cortex, shown in white, there are columns that receive heavy input from one eye, and they're separated by dark spots that presumably receive input from the other eye. So just like there's segregation in the LGN, there's almost perfect segregation within the visual cortex as well. And presumably the spots that are dark here, those receive inputs from the non-injected eye. So as we saw last time, there's 6 layers of the LGN, 3 representing one eye, shown in blue, the other 3 representing the other eye, shown in green. These project to the layer 4 of the visual cortex, but they do it in such a way that the layers of the LGN receiving input from one eye project to a patch within layer 4 of the cortex, the layers from the other eye to an adjacent patch, and so on, alternating across the cortex. Within layer 4, cells are driven from either the ipsilateral or the contralateral eye. But the cortex is organized in such a way that the main input from the thalamus goes to layer 4. Layer 4 then projects to the other layers, primarily the layers above layer 4. And as it does so, it spreads out a little bit. The result of this is that if you have an ocular dominance column, say for the right eye, if you record from layer 4 with an electrode, you'll just get activation from the right eye. If you go right above the middle of it, all the cells you encounter in the upper layers will probably be just right eye. But if you go a little bit off the middle, you're going to start getting mixtures of right and left eye. Do you see that? And if you've got a cell here, like right between the ocular dominance columns, that might be equally activated from both right and left eye. So in layer 4, it's right eye or left eye, ipsilateral or contralateral. But when you get into the other layers, 1 through 3 and 5 and 6, individual cells are different. Some are just one eye, some are the 2 eyes almost equally, some are mainly one eye and a little bit the other. And as I told you last time, if you drive an electrode through at an angle, you find that the eye from which they can be driven shows a nice, typical, neat alternating pattern back and forth. This is the work of Hubel and Wiesel, who won the Nobel Prize for it, and a lot of what we're going to talk about today is Hubel and Wiesel's work. We saw that you can actually visualize 3-dimensionally what these ocular dominance columns look like. We were looking at cross sections through the cortex, so it's like if you took a slice like this. But if you look at the cortex face on in surface view, I described last time how you could visualize the ocular dominance columns. So this is a cortex viewed, as it were, from the surface. And you can see there actually are stripes of cortex that receive input from the right eye. God bless you. Separated by stripes that receive input from the left eye, and so on across the cortex. And it's really remarkable because there's no hint of this complex organization in the conventional staining of the cortex, but it's there anyways. Now, here's an interesting fact. I didn't mention this last time. If you look at a normal individual, the right and left eye stripes are of roughly equal diameter. What do you suppose happens if you take an animal? This happens in humans too under some conditions. What do you suppose happens if you take a monkey and say for the first few weeks after it's born, you put a patch over one eye? Probably what you're going to find if you examine that monkey's acuity is they're not going to be able to see quite as well out of that eye as out of the other. And things like this actually happen in humans. In humans it's called amblyopia. Have you heard of that ever? A-M-B-Y-L-O-P-I-A. Amblyopia. A-M-B-Y-L-O-P-I-A. That refers to the fact, the situation where there's a visual impairment in one eye, usually in children. And if that visual impairment is not corrected early enough, the child might never have completely normal vision out of that eye. I actually think I have very mild amblyopia. I started wearing, for me, my left eye for many years was basically normal. I could see just fine out of it. But my right eye was extremely farsighted. So I started wearing glasses in second grade, and the left eye was just window glass. I didn't need anything there. But the right eye corrected for this farsightedness. And so even now, with my glasses on, I'm aware that if I close one eye and close the other, I'm like aware that I can see just the same out of both eyes. But if I try and read through my right eye, it doesn't flow smoothly somehow. It's like, and I can't say why. I can see the letters and words. It just doesn't fit together. So I think I have a very mild amblyopia in the right eye. It could be more severe. Maybe you've heard of some children who have a squint, whose eyes don't line up, and they'll actually patch the weak eye. Have you seen that ever? And they'll do that to prevent the development of amblyopia. So amblyopia has been known for ages. There was, so yeah.
So why can't it be corrected later on in one's adult years?
Well, I think I may be able to answer that in a second. The answer is it just can't. It can be prevented from occurring in a child. And as I said, one way is just to put a patch over the normal eye for a certain number of hours a day to force the child to use the good eye. But some experiments I'm about to describe might explain why this amblyopia occurs. And these are really just incredible results, I think. So normally the right and left eye stripes are about the same thickness in the cortex. But if you take a monkey and you patch one eye 24 hours a day, so it can't see out of that eye, and then, I'm not sure how long you have to wait, but you wait a certain period for development. If you test then, the animal has very poor vision in the covered eye. And I didn't bring in a figure of it, but if you do an experiment like this, what you find are very thick stripes corresponding to the normal eye, and very, very thin stripes corresponding to the patched eye. It's as though the cortex doesn't receive enough input from the patched eye, and the part of the cortex that normally would be used to analyze that input is suppressed, and it's given over to the other eye instead. So the reason for amblyopia may be there's nothing wrong with the eye that's not correctable by glasses in most cases, but it may be that during development, the cortex gives most of its space to the eye that works right. So as a result, there's just not a lot of cortex left over for the patched eye, and so the animal can't see normally out of it. And this is very likely to be the reason for human amblyopia as well. The retina, the eye could be corrected with glasses, but there might still be a problem. Okay, that's where we got to before. Let's press on quickly from there. I have mentioned in every SED system that topography plays an important role, correct? And the visual system is topography in its most perfect form. Different parts of the visual field are represented in different parts of the visual cortex. And this was actually first studied carefully after the First World War. There were so many thousands of soldiers who had penetrating wounds, usually bullet wounds, that involved the occipital cortex. And if they were examined carefully, they had scotomas. Remember that word? They had blind patches within the visual field. And it was fairly soon realized by neurologists that where the blind patch was correlated with where the bullet or bayonet or whatever had entered their brain. So there were actually two very famous studies, one in the US and one in Japan, comparing visual field deficits and the believed location of damage. And as a result of this, they were able to come up with a very precise map and say this region of visual field is represented in this part of the cortex. And they were able to very beautifully work out the details of cortical topography. More recently, you could demonstrate this pretty easily using fancy imaging techniques. So here's a result of some human imaging. The person was asked to fix on a point. Now these are false colors, don't get confused. The colors are just to illustrate things. The person looked at a point, and they then put dots of light in various regions around that point. If they put a dot in this red region here, they got excitation. This is a functional magnetic resonance image is constructed from. If they had a light in the red spot, they got activation somewhere within that red region of cortex. If they put the light within the green area, they got activation within this green part of cortex. They put it within the blue area, they got activation in this blue part of cortex. So this is one way of demonstrating the topography. And there's one thing I want you to note here that's very, very important. Look at this central area. That corresponds roughly to the fovea. The fovea is about 2% of the visual field. It's real small. Look how much of primary visual cortex, look how much of area 17 is given over to the fovea. It's almost half of it. So the fovea is a few percent of the retina, and yet it's more than half of visual cortex. I've discussed before how you can really see so much better in the fovea when you're looking right at something than elsewhere. And you would see now there are probably a couple of reasons for that. One is that high density of cones within the fovea, but another one is much more cortex is given over to the central part of the visual field than the peripheral part of the visual field. It's like the center part of the visual field is what's really important to you. And so the brain uses most of its resources for that. It's just like, I'm sure Dr. Carr talked about this. If you look at somatosensory cortex, like your fingertips are a tiny little part of your body, but they're a huge part of somatosensory cortex. Your fingertips are really important to you to touch. And so the brain devotes a whole lot of space to the cortex. It's the same thing in virtually every sensory system. That whenever there's a region of sensory space, which is exceptionally important, the brain devotes a excessive amount of space to analyzing that. And that's just what we see here. You can actually beautifully see the cortical magnification. That's the word they always use. You can see the cortical magnification. That's a gorgeous figure I actually started talking about last time, but I think sadly I'm going to skip it because we've lost I'm sorry. We've lost a whole bunch of space. And we have even more important, even more exciting things to get onto. We've discussed a little bit about the receptive fields of cells in the retina, correct? And we saw that in general, a retinal ganglion cell has a circular receptive field containing two concentric areas, either on center, off surround, or vice versa. Remember that? Let us now, ladies and gentlemen, press from the retina into the brain proper and see what happens. First of all, let's look at the lateral geniculate nucleus. And this is pretty simple because the LGN cells have receptive fields very, very similar to those of retinal ganglion cells. So within the LGN, you find two main kinds of receptive fields. You got on-center fields with off surrounds. So you shine a light on them, you get a response. Shine a light on the periphery, you get a suppression, just like we described in the retina, right? Conversely, if you look at an off-center cell, it's suppressed when you shine a light in the center, and it's excited when you shine a light in the surround. It's not shown here, but in a lot of these cells, if you shine a light on the suppressive region, very frequently they'll show a little burst of firing when you turn the light off. It's not always seen. These cells show pure suppression. They just turn off here and here. But real often these cells, if you shine a light here, they're suppressed. When you turn the light off, they'll often have a little tiny burst of firing just as the light goes off. Same thing in the retina. Okay, makes sense? Just like the retina. Nothing new under the sun. However, ladies and gentlemen, when we go on into the cortex, things get very different. And this difference might not seem to be that big a deal at first, but I will attempt to show you that it actually is, in part today and in part on Friday. There are several different types of receptive fields found in visual cortex cells. So that's unlike the LGN, where they're basically all the same, like I just described. There are several different types of cells found in area 17. The most common, or the most famous type, is the so-called simple cell. Here's a receptive field of a typical cortical simple cell. You will note we still have the eccentric on and off regions. So this cell has an excitatory center and inhibitory surround. You find the opposite as well. I'm going to stop saying that every time because it's always true. So in that ways, it's just like the retina or the LGN, but what's the big difference between this and the cells we've looked at so far? The shape. They're no longer round, they tend to be elongated. Now that might not seem like a super big difference, but let us think, ladies and gentlemen, about what the optimal stimulus for exciting this. What's the best stimulus for exciting a on-center LGN cell? It would be a dot of light that just fills it, correct? What's the best stimulus for inhibiting it? It would be a round ring of light that just surrounds it. But how about a cortical simple cell? What's the optimal stimulus? Can you see it? What's the optimal stimulus for exciting this cell? It'd be a bar of light, would it not? It would be a bar of light at this particular angle or this particular orientation. Can you see that? The more of the excitatory center you fill, the bigger excitation you're going to get. Here's some real data just to show you I ain't making this stuff up. Here we have a cortical simple cell, and these are its responses to various stimuli. Here's a bar of light placed at this angle in the middle of the receptive field. You get a nice excitatory response. Here's a bar of light placed not in the center, but placed in the inhibitory surround. There's no response to the light turning on, but in this case, when it turns off, you get a little burst of firing. Sort of like I described sometimes happens in LGN cells. Here's a nice bar of light, but it's at the wrong orientation. Can you see that? What's that cell going to do? Well, it's probably going to do nothing at all, because these cells are sensitive to the orientation of line segments. Basically, if it's in the right orientation, in the right place, you got a good response. If it's like a little bit off, see, then you're getting some of the excitatory center, some of the inhibitory surround. It'll be suppressed, and when you get far enough off, the response will disappear entirely. This is if you just turn the light on entirely, and you get virtually no response at all. That makes sense, because you're getting the center and the surround. And diffuse lighting is a bad stimulus for the retina and for the LGN as well. So superficially, this elongated receptive field might not seem like a super big deal, but you can see it results in cells with very different properties. These are cells now that like lines at certain angles. Now, think about that for a second, and maybe you can see why this is such an important difference. Think about the world around you. The most important thing for the visual system to detect, and remember like what Dr. Hilbert was talking about, the goal of the visual system in one way isn't just to transmit light into the brain, it's to find out about the world outside, right? And what's the most important thing that tells you about the world outside of you? It's boundaries and edges, is it not? I mean, that's why you can draw things with a pencil, where you just draw the outlines and you can still understand what you're saying. Cells like this detect lines. They in fact are line at a certain orientation detectors. So in a way, this is a big, big change from the spot detectors that are represented in the retina. Now, a question that arises is, well, how do we get from those round receptive fields to these elongated receptive fields? And Hubel and Wiesel proposed a model, and it's probably basically correct. Their model ran like this. Now, I found that people have trouble understanding this, and it's one of these things that's really not that complicated. Maybe it's just difficult to explain. So I'll try and explain this clearly. And think about it, and if it doesn't make sense to you Friday, I'll try it again a different way. Sound good? So I'm going to try once. It's your job to determine whether I've done it or not. Here's the lateral geniculate. Here's 1, 2, 3, 4, about 5 or 6 cells. Schematically, this represents the receptive fields of a bunch of cells in the lateral geniculate. So in this case, remember every LGN cell has a little spot somewhere in the visual field from which it's excited, right? So in this case, we've taken, I guess, 4 or 5. I'm not sure if that's a cell or not. We've taken 4 or 5 cells. These circles with the X's mark their, or pluses, I guess, mark their excitatory centers. So these are a few cells that just happen to have their excitatory centers lined up in a straight line at just this angle. Do you see that? So suppose you feed all 4 of these guys, here they are schematically, suppose you feed all 4 of these guys into one cell in the cerebral cortex. Now, cortical cells are pyramidal cells, so they really should have shown this triangular in shape. I was so irritated when I noticed this the other day that I was tempted to redraw it, but I was too damn lazy, and you're just not worth it. But, but I hope you get the idea. These are cortical pyramidal cells. But understand what's going to happen. If you have light at just the right angle, it's going to hit the excitatory centers of all these guys. All 4 LGN cells are going to fire. All 4 of them are going to shoot into the cortical cell. That's probably going to result in that cell firing vigorously, right? Suppose you had a line at another orientation like this one. Well, that's only going to hit the excitatory center of one of these cells. Can you see that? As a result, there's just going to be a very weak excitatory drive to this cortical cell. If you're off-center, well, you're missing all of the excitatory centers, and instead you're hitting the inhibitory surrounds. A lot of LGN cells have a baseline firing rate. That's going to result in no excitation at all, maybe even inhibition of the cortical cell. Does that kind of make sense? And again, this is almost certainly what's going on, but if you think about it, you got to think how complicated this is. Millions of cells in the LGN have to connect themselves to the cortex in just the right way in order to get these cells to have this line detection ability. If you took 4 or 5 random LGN cells and connected them to the cortex, you're just going to get a mishmash. There must be some developmental mechanism that causes groups of cells, LGN cells, whose centers are lined up the right way to connect to individual cortical cells. So there's still mysteries about just how that might occur. But does the basic mechanism kind of make sense? Now we could spend all day with no problem talking about cortical simple cells, but let me just demonstrate one other property of these cells. This is not universal, but it's true of a large number of cortical simple cells, and that is that they are sensitive to motion. They're the best stimulus for exciting them often is not a stationary stimulus, but a moving one. And this fits with the fact that in general, our visual systems, and that of almost all animals, really like movement. Our visual system is very tuned to movement. I have a pet lizard. Got her for my daughter when she was in 6th grade. Here we are 18 years later. She hasn't fed the thing in 18 years. I just can't bring myself to let it go. But I got to admit, I don't feed it regularly. So it goes through, what's the big deal now with losing weight? Intermittent fasting. This lizard has gone for 18 years of intermittent fasting. And this is way longer than they're supposed to live. So I think it's been really good for her. But I feed him crickets, and the cricket's moving. It's like, wow, it gets his whole attention. The cricket freezes, and it's like he doesn't know where it is anymore. And this is characteristic of a lot of animals. Even yourself, it's night and you can't see well, there's something moves, that gets your attention, does it not? Your whole visual system is really tuned to moving stimuli. And so these simple cells are especially tuned to moving stimuli. What's incredible is many of them care not only about the stimulus moving, but about the direction in which it's moving. So here's a recording from a series of recordings from a single simple cell in the cortex. And the stimulus here is a bar of light, which is moved back and forth. So you move the bar of light back and forth like so, cell doesn't respond. Put it up and down, bar of light's like this, doesn't respond. Put it like this. This is what it does. Put it like this at a little bit of angle, and the cell responds like crazy. But note, it responds only when it's moving to the up and right. See, it doesn't respond at all when it's moving in the opposite direction. So this is a cell that to get the maximal response out of it, not only do you need a bar of light at the right angle, but it has to be moving in the correct direction. We don't have time to go into this, but it turns out that circuitry has been worked out that's able to perform this relatively simple kind of computation. So that's your typical simple cell in the cortex. It's orientation selective, and not always, but often movement and direction sensitive as well. Now, if there are simple cells in the cortex, I think you can guess there got to be complex cells as well, correct? And indeed they are. And this is the typical responsiveness of a typical complex cell. Here roughly is its receptive field. They've drawn it as a square, but most of these are really ellipses. It's just easier to draw them as squares. Let's see. Here's a stimulus, a bar of light there. Do you see it? Cell responds pretty well. Here we move the bar of light over a little bit. Cell responds pretty well. Here we move it over a little bit more. The cell responds pretty well. Here we put it right across the receptive field at another angle. No response at all. How does this differ from simple cells? There's no longer a concentric center-surround organization. Complex cells require a bar of light at the correct orientation, but the bar of light can be anywhere within their receptive fields. Some of them, again, are motion sensitive, but the main thing is in complex cells, there no longer is a concentric center surround receptive field organization. Some of them, not all of them, some of them are said to be end-stopped, which means there's an optimal length to the bar of light. Some aren't, like this is a non-end-stop cell. In an end-stop cell, if it went from like here to here, you get a nice response, but if it stuck out over the edges, you wouldn't. Some complex cells are end-stopped, others aren't. But you can see there's a change now. We've gone from a bar of light at the exact right orientation, at the exact right place in space, to a bar of light sort of like anywhere in this general area will be sufficient to set the cell off. And again, the topic of modeling how complex cells are constructed is a big topic. Hubel and Wiesel suggested that complex cells, so we saw how simple cells could be made out of LGN cells, correct? Hubel and Wiesel suggested that complex cells can be manufactured from simple cells in the same kind of way. That if you have a whole bunch of simple cells with the same orientation and feed them into a single cell, that will generate a complex cell. Does that sort of make sense roughly? So that was the Hubel and Wiesel theory. Unfortunately, it's almost certainly wrong. It's a shame because it's a beautiful theory, proved wrong by some ugly facts. If that theory was true, if first you had simple cells and the simple cells connected up to give you complex cells, that would mean the response latency of the simple cells would have to be less than that of the complex cells, right? If light goes from the LGN to simple cells and from simple to complex, the simple cells would have to fire first and the complex cells a tiny little bit later. Sound plausible? But it's not true. There's no difference in latency between the response of simple and complex cells. So they're not generated in this fashion. It must be something about the way the LGN projects them. It's not understood yet how that happens. We've seen that cells in the cortex sensitive to the right and left eye. Yes?
Which do you have more of, the complex or the simple?
I think they're more simple. But not by a huge amount. There are a lot of both. We saw that the right and left eye sensitive cells are not like randomly scattered around, right? They're arranged in columns. And you might wonder, does something like this happen with simple cells, with simple and complex cells in the cortex? Actually, for lack of space, let me just go to this picture because it's prettier. This is an image, fairly recent, or not that recent, 92, of, thought it was newer than that, of monkey cortex. And what they've done is the monkey was anesthetized, and it's a striking thing that all of us, the retina, the LGN, primary visual cortex, they all work just fine in the anesthetized animal. It's a remarkable thing, because when you're anesthetized, you don't consciously see anything. But clearly the anesthesia is not working by suppressing these early stages of visual processing. This is a study in an anesthetized monkey. They removed some bone to expose a window over visual cortex. They then put a voltage-sensitive dye over the cortex. This means that if a bunch of cells fire in one place, the dye will change color. Does that make sense? So doing this, they put line segments at different orientations. This is just fantastic stuff. They put line orientations at different segments in front of the animal, and they examined literally which areas of the cortex lit up. Now this is false color. This isn't what it really looks like. But what they've done is, do you see these little dashes on here? Those are the orientation of the line segment. So look at this blue area and see how the dashes are going like this. When they exposed the animal to bars of light at this orientation, it turned on all these blue areas of the cortex. Now each of these contains hundreds or thousands of cells. In other words, cells respond